something completely different. We are talking about an Icelandic biotech company, which I don't think you will come along that often, but at least I have one. So this entire project started in the early 2000s by a number of pulmonologists in Iceland that had COPD patients. And they also have, you might know, the aluminum smelters. So they have a lot of lung patients from those smelters as well. And these pulmonologists were quite confused because they realize that the only thing that really works for COPD is to prescribe the patients azithromycin. Now, azithromycin is an antibiotic, and of course these patients often suffer from infections, but what is really weird is that azithromycin is the only thing that works really good for COPD if you don't have an infection. So they started to dig a little bit deeper into this and kind of figuring out what's really happening here. At the same time, pharma industry continued on the pathway to develop more and more anti-inflammatory agents for COPD. So far, that hasn't really been successful, and I personally don't think that if you develop yet another anti-inflammatory agent, it will probably not solve the COPD disease as we know it today. So what they come up with was actually they started collaborating with the university in Reykjavik, and they could show that azithromycin actually had two different mode of actions. It was antibacterial for sure, and it's very clear how that mechanism works. But they could also show that the compound azithromycin enhances the barriers. So if you take biopsies, lung biopsies, and grow them on an air-liquid interface, and treat the liquid phase with azithromycin, you can see in a microscope that the cells are thickening, they are growing, they are maturing. You can also measure the osmotic flux over the cell membranes, and you can also measure the resistance over, and you can see that they are really maturing and become really tight epithelium. So then came along a really weird MD student who said, this is great, let's make a drug out of it. And suggested that if you take all these good activities of azithromycin and just remove the antibacterial effect, we should probably have a new drug. Which they did. So we now have what we call a new set of compounds. We call them the barolytes, that actually shows the exact same effect as azithromycin in vivo, but they don't have the antibacterial effect anymore. You know what, let me go back to this one. Who changed my slides? Never mind. So this is a busy slide. Let's see if we can take it from the middle and then work our way outwards. If you see the normal integrity, you'll see what is supposed to be a healthy epithelium, lung epithelium. What you see is that there is no flux over the membrane. Now, if you have COPD, you don't really have a genetic disease. You don't have a mutation somewhere that causes a disease. Most probably, if you have COPD, you're either a frequent smoker or you live in a polluted area. So what will happen is then in your lung, you will have a lot of free radicals, you will have a lot of acid, you will have a lot of particles. And what they do in your lung is that they actually physically break the lung, the barrier. And what then will happen is, if you look at the high TOI, later down the middle, is that they will cause an actual physical leakage on your lung. So what you're having, so you will have a leakage in both ways. So what's in your lung will go into your body and the other way around. And this is then when you get diagnosed with COL or COPD. Of course, the body's natural response to this, if you have a physical damage to your lung barrier, is that it will form an inflammation. It will start sending out granulocytes and macrocytes to get this. And of course, this is where we think the misunderstanding comes from, that this is an anti-inflammatory treatment. But we believe that inflammation is just a symptom. It's not really the cause of the disease. So if you look at the triangle, the pyramid to the left, you can see that we are thinking that COPD for us is not an anti-inflammatory disease, it's not an infectious disease. This is a case of a disrupt barrier in your lung. And if you can then heal the barrier, you will have then a better treatment for COPD moving forward. So far, we have raised 2.2 roughly million euros, mainly by local fishermen on Iceland, because they have a lot of money. Iceland is a quite closed society. They are actually all relatives, quite close one, sometimes too close. So we also had received some grant funding. Again, it's their family who sits in the grant committee, so that helps. And we are here now looking for more finances so we can actually take us all the way through phase 1. What they don't really have a lot of in Iceland is people with a biotech background and experience. So they brought me into this company in April this year. So I'm from Sweden with 20 years experience in pharma, and we also have the chairman of our board. He's here in London, Clive Page, at the Sackler Institute, to get some more know-how to the drug development side of this. If you look at the top right level here, I think this comes back later, we'll show it again, is that the way that we think that this is not an inflammation, it's a barrier leakage, kind of limits the amount of available animal studies that you can do. Because as soon as you call the usual suspect, the animal houses, they are very happy to learn that you're in COPD, and they send you a long list of animal studies that you can perform, all of them measuring the anti-inflammatory effect of your drugs, which is not really what we're after here. So we had to develop our own setting, and Icelandic people, if you have met them, they are quite straightforward. They don't really mess around so much. So what we do is that we're actually pre-treating our mice with either azithromycin or our compounds for a few days, and then we basically put them in a gas chamber and expose them to sulfur dioxide gas for a few hours. And of course, what that does to the mice is that it forms sulfuric acid inside the lungs, and of course, then by the acid, it actually destroys the epithelium. Now, how do you then measure the leakage, the epithelial leakage? One way in animals, it's not practical in humans for obvious reason, is that you inject human serum albumin in the tail of the mice. You kill the mice, and then you flush the lungs with fluid, and then you can measure how much of your human albumin is now in the lung fluid. And that's a good measurement of barrier integrity. And we can see that if you have the untreated, the PBS-treated mice, that we normalize the damage of the lungs. We can see that we have 40 to 60% reduction of lung breakage or lung leakage with our compounds, or with azithromycin for that sake. Now, of course, this is not really feasible to do in humans. So there there is a professor in Rochester, upstate New York, who is developing an alternative diagnostic tool for COPD. There is the gold standard that we are using now, and he's developing another one. He lets his patients breathe mannitol, and then he takes out the blood samples and measure the level of mannitol in the blood. And that is also then, again, a measurement of your barrier integrity. And he do this because he wants to find another classification system for the epithelium integrity. Interesting enough, he is only quoting his research is only quoting the Icelandic papers from the early 2000s. So he took their invention and kind of put it one spin on it, and we're taking another. He is also very kindly developing our diagnostic tool. We haven't told him yet, but we will eventually when he's done. Of course, the organization is completely virtual, I would say. We have a few scientists at the University of Reykjavik who actually performs all the hands-on, all the in vitro and in vivo studies. And then we are a group of consultants running this. And of course, we are using the usual set of suspects for all our studies. So the purpose of me being here today is to raise another 6 million euros. That will take us all the way through phase 1. And we will do the phase 1 with an actual extension arm. So we will have, first we'll do the regular SAD-MAD study, as everyone else. And in the end of that, we're going to take COPD patients, let them breathe the mannitol, treat them for 3 to 4 weeks, and let them breathe the mannitol again, just to see if we're having any improvement. The COPD study itself is, when we're going to do the phase 2, is quite long, but this will at least get us some sort of feeling whether we have some kind of signal on barrier integrity or barrier enhancement. So I think that's pretty much what, well, wake up. Do we have any questions? I'll be around for another few hours if you have any questions afterwards. Okay.
The mechanism, what extent have you understood that?
Yes, so we see it on the microscopes that the cells matures, that they tighten. We don't know the actual target yet. We are working quite hard on it in Iceland on genetic expression. We're looking at the mRNA expressions, up and down regulations, and we're doing some other mechanism of action studies to see what's going on. So far, the only thing that the thing we're building the case on is that everything we do, we compare to azithromycin, because azithromycin works in patients. So we try to see that whatever we do, we always try to mimic or outperform azithromycin.
So have you managed to outperform it?
Yes, mainly in the case of oral bioavailability and PK profile. So we've done a lot of those studies. We see we have a better PK profile. It's orally very bioavailable. We have taken the lungs on the mice and we have dissected them, and we have actually measured the drug concentrations in the lung. So we know it actually hits the target organs, not just it goes out in the plasma, it actually reaches the lungs where it should act. Of course, there is a fallback strategy. We can always do an inhaler if we are worried. But so far, I think a once-a-day oral, if you have this COPD, you're going to have it for a long time. So that's the fall. But we're working on it. Ask me again in a year. Thanks.